So first of all, I want to apologize. When I revise abstract, I forget to change the title. So I will not describe a bunch of troublemakers. I just stick to one, Nosema. The Nosema is a microsporidium parasite. It's single cell, no mitochondria, spore forming, and intracellular. It means all the energies it requires to reproduce from the host. The infection cycle is about 4 days. It starts from the ingestion of spores. Then the spore germinates. Then it germinates and then enters the epithelial cells. So it infects epithelial cells of the middle gut. After 4 to 5 days, a lot of offspring spores will be produced. And then the cell will burst. So the new offspring spores, they can go to the environment to infect the new individual bees. They also can germinate immediately in the midgut to infect neighboring health cells. When we discuss or talk about host and parasite co-evolution, we have to consider both sides, both hosts and parasites. The adaptation is mutual. So how the host was selected to adapt this new parasite and how the parasite adapts this new host. Then can we control it with siRNA? I studied from the host part. It's easier. So there is a selection program in Denmark for a very, very long time. The Nosema was not found there. So supposedly there should be tolerance. Then I do some mortality tests, infect bees from Denmark and somewhere else. Then great, they have very or super low mortality. Then I do a classic QTR mapping, then cross to select the strain and unselect strain to do QTR mapping. So the genotype is microsatellite markers, and the phenotype is how many spores the offspring can produce. Then here is a chromosome of honeybee, 16 chromosomes, and the chromosome 14, we find a signal which suggests on chromosome 14 this region could be selected for the tolerance. If this is true, we should be able to find the selective footprint from the initial population because they are the bees under the selection. What can I do? Probably compare the genetic diversity. So let's check the diversity of the original selected population and collect some bees from the wild to see if they are under the selection, the diversity should be super low. If they were not, then that means our QTR mapping results might be wrong. Zero means the diversity is the same between the selected bees and unselected bees. When the value goes to minor, which means the diversity of the selected bees are lower than the unselected bees, what we find is a select sweep from the QTR mapping region. That's exactly what we found from the QTR mapping. The number of alleles is 1.1. That suggests this region is fixed. That is really under selection. Then the next step, we have candidate regions. Great. So then let's go for some candidate genes. There are 31. Then compare their protein sequence with the Drosophila and the human. That's one super interesting. That's aubergine. That is involved in RNA interference. Particularly in Drosophila, that is involved in attacking the foreign pathogens. Then we say there are two ways they could be functional. One is the selected bee have a new allele. So the sequence is different. That makes them more strong. Alternatively, they could be different at the expression level or both. They have a new allele, they also have a high expression level. I didn't sequence them, so I cannot tell whether they have a new allele, but I do the qPCR to see the expression level is much higher in the bees, has low spores. This candidate can be true, they can be wrong. I didn't do RNA confirmation. Irrespective is true or wrong, it has strong impact for my follow-up study because of this candidate gene. Then I go to the parasite part, the Nosema ceranae. I start from the easy way, do some RNA sequencing, check the expression. What I do, I didn't do it for 1 day, I do it for an entire infection cycle from day 1 to day 6. So make sure I can cover all different reproduction phases of this parasite. mRNA sequencing is easy, we do gene expression. Why do microRNA? Because I get a candidate from the QTR mapping that is involved in RNA interference. That's why I do small RNA sequencing. Here's the graph. X-axis is day 1 to day 6. Y-axis is the percentage of parasite gene was expressed. So in total, there are around 2,000 genes for Nosema ceranae. And on day one, like 25% was expressed. So they were not expressed at the beginning, all of them. They will have selections over and over and over. Around day 3, most of them are expressed. They are fully expressed. Are they, for those genes, they are evenly expressed, or they have specific pattern, highly expressed at certain points, then they decrease? Yes, it is. There are 4 different patterns. And for the genes coding DNA polymerase, RNA polymerase, ATPs, those involved in reproduction was highly expressed as the initial part, day 1 and day 2, then they decrease. For Nosema ceranae, there are 2 hypotheses suggest its violence. They are non-exclusive. The first one is they can secrete hexakinase to the host cell, then accelerate the metabolism to support the parasite reproduction. The other one is the infected cell cannot start apoptosis. From the first hypothesis, what we find is hexakinase is really highly expressed from 2 days post-infection, keep high expression. So we cannot compare to another treatment. What we can say is they are highly expressed after they enter the cell. Then that's all. Then what we do for the B part, as we use in the mid-gut tissue for the sequencing, so we can harvest the information both host and the parasite. Then for the parasites, is first reported on 2006. What I did, this is 2013, around 6-7 years. How could this parasite adapt the infection? What I can think or I can imagine is, at the beginning when the parasite comes, diversity is high. After many years selection, the diversity should be reduced to local adaptation. Here, x-axis. Here is an example of conical 1. X-axis is the physical position from 0 kb to 50 kb of first conic. This is the longest conic of the assembly. Y-axis is the number of SNP for each open reading frame. So the black dot is the number of SNP we can find in 2007. And the red circle is number of SNP we find in 2013. It suggests every open reading frame, the number of SNP is reducing. They are losing diversity. After finding this decrease of SNP, those SNP really change the protein. So we test this for the synonymous and an unsynonymous one, which means the selection is really at the protein level. They are really changing the protein. That's why we suggest a purifying selection. After parasite, then is the host. From day 1 to day 6, we do gene expression study. It's not surprising to find hundreds, even thousands of different expressions. That's what the RNA sequencing do. We can find on day 1 they are certainly different. Day 2 different. We can pick our favorite ones for each day. Then good luck with that. It's really hard. So what I do is looking for the extremes. They were significantly regulated from day 1 to day 6. Always regulated. There are certain of them, but most of them are hypothetical. We don't know the function. But luckily there is one. This is the mitochondrial gene. It involved apoptosis. It was suppressed from day 1 to day 6. It means the cell cannot initiate apoptosis. So generally, there are 2 ways to start apoptosis. One is the blood cell. There's no report on the blood cell on the honeybee midgut. So the other one is mitochondria. It feels very rare. This is a mitochondrial gene involved in apoptosis, and the cell cannot die. Then I do small RNA sequencing. It's not surprising one microRNA regulates many mRNAs. And the single mRNA was regulated by many small RNAs. What I do is first test how many microRNAs of the honeybee was significantly regulated by this infection. Then I get them, predict their targets. Only predict, I cannot make sure, I cannot confirm. So now I use their seed region to the 3' UTR of the mRNA for the predict. For those 70 microRNAs, around 400 honeybee genes could be targeted. Let's say if they are really targeted gene, we should be able to find correlations during all 6 days. Another thing is if those microRNA are targeting genes that has completely different functions. And what we have found is a lot of different co-expression patterns, co-expression groups. Here we have only 2 co-expression groups of these targeted genes, means their functions are very similar. And also what we found is from 7 out of 9 pathways from these genes are metabolism related. That means the microRNA of honeybee respond to Nosema ceranae infection, and the direction of this response is metabolism related. Now we have microRNAs of the bees. Is there any microRNAs of the Nosema? So far we know there are 2 RNA interference orthologs in Nosema ceranae. One is dicer, one is argonaut. There are also similar genes annotated from other microsporidium parasites, but there is no functional analysis. Nobody knows whether they express, whether they are functional. From the gene experience study, we know these 2 orthologs do express from the Nosema ceranae genome, but are they functional? If they are functional, we expect some microRNAs. Since we already have the reading, the sequencing data, it doesn't cost anything to do some predictions. So with prediction, we indeed predict more than 6. Then we need to confirm. So design the primers to test the existence of these true microRNAs. What we expect, or what I expect is, I should have positive results from the infected group. That's great. I have product. And I do not want to find any product from the uninfected control. That's it. So the take-home message is there is microRNAs from Nosema ceranae genome. But which genes they are targeting? I don't know. They might target their own genes for the reproduction regulation. They could also target the host gene to change something for the bees to make them able to reproduce. What we can do? We can do RNAi to silence one of those microRNAs. That will be a lot of work. I don't want to do that. At least not at the moment. What I do is I target the genes that's important for the small RNA maturation. That is 3 genes, one from host origin, I get from the QDR mapping. The other 2 is from the parasite, is dicer and argonaut, with different dosages. On this graph, x-axis is the treatment. Y is number of spores. They are diluted. We were not able to count 10 spores from a bee. When the bees was targeted with the dicer, we found very, very low spores from this treatment. That is exciting, but I have to watch it carefully. Maybe I'm too tired, I can't run, or I didn't mix the solution properly, then I can't run. So I want to confirm. The first confirmation I do is qPCRs of dicer. Since I'm targeting this gene, the expression level should be low. If they are high, then I'm wrong. If they are low, great. Good luck for me. I feed the siRNA for dicer for 3 days. For the first 2 days, the expression is super low. Day 1 is undetectable. Day 2 is still low. On day 3, there is not much difference. It's much because when decrease the sufficiency of siRNA pathway, this site is not functional anymore. So the siRNA pathway is not functional anymore. They cannot suppress it. Still, I want to confirm it. I repeat exactly the same experiment. Then I have the same results. Thank you.